how they inspired, sorry, and how they are still inspiring fundamental and biomedical research, but also bioengineering, design, and architecture. So, the Craniata is a proposed clade of chordates that you already know, and which we are part of. So the Craniata are defined by their skull, that can be in hard bone or in cartilage. And their marine representative are the Agniata, which are the hagfish and the lampreys. And so Agnata is without a jaw, so a jawless fish, and the Gnatostomata. So every vertebrated with jaw. So they are composed, Gnatostomata are composed by bony fish, cartilaginous fish, but also tetrapods. So the marine tetrapods are seabirds, reptiles, reptilians, and mammals. So we are going to show you that marine craniates, from this organism to this one, can inspire so have inspired human in many ways. But first, what is bioinspiration? So the definition that we found is that bioinspiration is using solution found in biological systems or biological evolution to develop innovative material, devices, and structures. But we think that bioinspiration have to take into account a more sustainable way to exploit the marine potentials. So first, we'd like to talk about a pretty interesting molecule. Actually, you know that some fish live in extreme environments, such as the polar cycle, and how this organism survive intrigued scientists for a long time. And in the late 60s, they found, so a group of scientists isolated the antifreezing protein, so the AFP, in the Antarctic rock cod, so this guy here, and so the AFP, they showed that the AFP was contained in the blood serum, and it is what that allowed the fish to survive in environment below 0 degree. So yes, they have two very interesting features. So it binds to ice crystals. So yes, they inhibit the growth of ice crystals, and they bind also to the cell surface to improve their viability. So they are now very studied in two fields of research: the cryopreservation for the organ transplantation storage, and scientists already went to a trial on rats, and there were promising studies where the vital organs of rats were able to be preserved much longer with this protein. But it's also being used in the food industry. So a few years ago, another group of scientists synthesized a designed compound, so they designed it themselves. It was based on the AFP, and it's called polypropylene, that is biodegradable and seems promising for both of these fields. But we also can talk about the importance of the lampreys in the evolutionary biology. So the lampreys are a member of an ancient lineage of vertebrates that diverged from vertebrates a long time ago. And so as one of the only surviving group of jawless vertebrates, they have long been studied as a model for understanding vertebrates' evolution. So they are also used to provide perspectives on the evolution of development pathway that defines vertebrates, like embryogenesis, the nervous system of vertebrates, the genome structure, their immunity, and so on. So it's still used. And but there is a lot of other ways to that Craniata help the fundamental research. And for instance, the study of otoliths that we already talk about a lot in classes. So otoliths are found in bony fish. And among a lot of uses in science, the otoliths helps researchers to study fish stocks and to implement a new sustainable form of fisheries. Okay, that was for the fundamental research.
Thank you. So I'm going to talk about biomedical research, and we'll start off by talking about the shark skin. So shark skin is actually made up of a matrix of small scales called denticles, which have ridges on them, which are called riblets, which you can see here. So here's one individual denticle, and then the riblets are the raised parts, and then they form this matrix here. And the amount and spacing of riblets actually varies by shark species and also along the body of the shark. So this picture you have here is one shark, different places along that shark, so like the tail, the head, the middle. And the texture of riblets, or the shark skin, actually helps resist fouling. So it's hard for microbes to attach to it because of this interesting topography that it has. So this has proven to be really useful for medical settings, like hospitals, where bacteria infections and bacteria spread is a huge problem. So one company, Sharklet, decided to investigate shark skin to propose a material, a surface that they could incorporate into hospital settings. So what they did was they actually took an impression of shark skin, and then they discovered that it has this diamond pattern with the kind of valleys and the ridges. So they replicated that here, and then they applied it actually to a catheter, which is one of the most commonly used and also most commonly infected pieces of equipment used in a hospital. And then they did some studies to assess this and found that it had a much lower bacterial load as compared to smooth surfaces or a regular catheter or whatnot that you would use in the hospital. So that's kind of quite a big deal because you can reduce bacterial spread without adding any kind of toxic things or cleaning materials or whatnot. So that's one application of shark skin, and Bailyn will talk a little bit more about shark skin later on. Another important feature in sharks that's been studied in biomedical research is their immune system. So the immune system can be divided into two parts. The innate immune system, which reacts kind of immediately. It has immune cells that fight foreign objects. It uses barriers, so the skin, and it works right away, as I just said. And the adaptive immune system, which reacts a little bit later, but it's more precise, and it has memory. So there you'll see things like antibodies and antigens, which are something that sharks have helped study because the adaptive immune system is a novel feature in vertebrates, and it's believed to have evolved from our most common ancestor with sharks. So that's why sharks are quite an interesting model for studying immunity. So two examples that we can look at specifically are IGNAR, which is an antibody that has quite different properties than what you would find in a human antibody, which is because obviously sharks live in the marine environment. They have urea in their blood to kind of help them adapt to this environment, which means that their cells have to be quite robust and quite stable. So if we actually incorporate, they did an experiment where they incorporated principles from shark antibodies into human antibodies, and they found that they were able to increase the stability. So they were able to raise the melting point of the antibody by 10 degrees. And in addition, that also increased production of the antibodies, the human antibodies that were produced. So if you think about this in a context of drugs, that indicates that we could potentially produce drugs that are more effective and also longer lasting. Squalene is another type of antibody that we can talk about, and it's a new class of naturally occurring antibodies found in animals. And It's positively charged. So how it works is it attaches to the inner membrane and removes positively charged proteins, which prevents viral replication. And it's really effective against a wide range of pathogens. And so far, it's proven promising and non-toxic in clinical trials for ovarian cancer and lung cancer. And then speaking more about antimicrobial properties, we can also look at seahorses for this. Again, they live in this environment which is full of bacteria, and they have an intimate contact with all of those pathogens that live in the environment. So a study was done in 2015 where they took 5 species of seahorses, and they used 3 different extraction methods and tested them against 20 different pathogenic strains, which were both bacterial and fungal. And they found that they had a stronger antimicrobial reaction to the bacteria as opposed to the fungi, but still a response to the fungi. So, and they think that's because, again, of the constant contact that they do have with bacteria. So this just, again, goes to show that there's a lot of really interesting bioactive properties and compounds that we can find within Craniata.
So now we are going to talk about the Craniata and bioengineering. So as Megan said, the shark have a specific scale and riblets. So one example that we show is the aerofoils. The aerofoils are the base structure of airplane, as you can see in this picture, through the modification of their surface. So in this first example that we show, the researchers inspired of the shape of denticles to have to improve the aerodynamic performance in aerofoils. So they model the scale of the mako shark in 3 dimensions, and then put the same structure in the aerofoil. So the results of this paper show that using this model, they discovered that a set of denticles inspire surface structure to achieve simultaneous drag reduction of lip regeneration on the airfoil. And also, they saw that the lift to the drag radio improvement the comparable to the best report for traditional aerofoils. So the next example that we show is the swimsuit. The many swimsuit manufacturers have developed inspired in the denticles. So also they modeling the denticles, and they design creating artificial denticles, and the scientific show that these denticles are able to prove that the shark skin reduce the energy. So they create this kind of swimsuit, but this is banned from Olympic Games because the people saying that when the swimmers swim in the pool, they go very fast. So for this, nowadays, these swimsuits are forbidden. And also we can find that this kind of shark skin fabric could be useful for swimming and robots. So the third example that we show is the ship biofouling. It's the same. The research can create the same structure of riblets and scale of the shark. And they saw that with this irregular face of the scale, surface of the scale in the ships can reduce the biofouling, and it's easy. It's more easy to clean. So the next example of bioengineering is we use the hamfish. The hamfish have a slim, is a defense of the organism. And these species have formed a glandular secretion that the name is exude. So the properties of this glandule are that does not dry out, are flexible, are stronger than nylon, and expandable are natural and renewable. So here the research create the slim and put in many applications. One of those is with military use, auto industry, biomedical, safety materials like helmets and vests, and also they saw that replace petroleum-based materials like nylon. So the last example that we can show is with seahorse also. We saw that the shape, that the tail of the seahorse are unique and different than other like monkeys or tiger. So here in this example, the research saw this unique structure of the tail because have a L-shaped bony overlapping plates, as you can see here. So also they modeling in 3 dimensions the same tail of the seahorse, as you can see in this picture. And they saw that it's more flexible because they have this form, square form, are more malleable and have defense against the threats. So with this, they inspired to create robot whisper and medical device.
So another example of bio inspiration we would like to present you focus on architecture. And we have the example of this 93 meters towers, which is built in China and represent, in fact, a fish soaring above the water. And moreover, the external part of the tower is made of aluminum pieces that represent the scales of the fish. Which one? Oh, okay. So, then, then we get the eels, which are migrator fishes that can travel 1,000 kilometers a year. And for that, they need to spend less energy as possible. So they have developed a particular flexible body, which is able of rippling through the water. And this ripple is what propels the heel forward. So this movement is known as the Van Karman vortex. So the bioinspiration here is to mimic this movement to produce energy. And the idea is to place a flexible membrane in the strong current zone that will mimic the movement of heels thanks to the force of the stream. And what will generate energy, and that will generate energy, and the company Hill Energy has already created a prototype. They function pretty well. And they want to, they plan a large scale production in 2020. The last bio-inspiration is the example of the boxfish and the automobile market. According to Mercedes, the boxfish has a particular shade that gives him a strong stability through the water. And it is exactly what they wanted to mimic with their new car concept launched in 2005. So Mercedes have developed a new car with a huge communication campaign saying that their new car is so comfortable to drive because of its stabilization design inspired from the boxfish. But the problem is that a study published in 2005 found out that the claim behind Mercedes inspiration were indeed false, saying that the shape of the boxfish doesn't seem to play a role in the increasing stability of the fish. So here is a good example of a bio-inspiration failure. So to conclude this presentation, I would say that Craniata is a clear example of clades with various bio-inspiration application. And it's time to change our perspective on animals. They aren't just for eating or looking at. We can really be inspired by all of their diversity and use it to find some solution to several different fields. Thank you.